The stages of meiosis. Before meiosis can occur, the cell must replicate all its DNA so that it has enough to divide into 4 haploid cells at the end of meiosis II. In humans, at the end of interphase, a cell would have 23 pairs of replicated chromosomes. In this model, a diploid cell with replicated chromosomes has 2 pairs of replicated chromosomes at the end of interphase. It is now ready to start meiosis. In prophase I, the chromatin condenses into X-shaped chromosomes. The nuclear envelope begins to break down. Spindle fibers begin to form from the centrosomes. Homologous pairs, sets of chromosomes with the same genes on them but potentially different alleles, begin to pair up. At this stage, crossing over could occur, where one section of a chromosome switches with the corresponding section on its homologue. In metaphase I, homologous pairs of chromosomes line up together at the metaphase plate in pairs, but the orientation of each pair is random. This is one method of creating genetic diversity. Note that this is different from mitosis, where chromosomes line up individually and not in pairs. Spindle fibers begin to attach to the chromosomes. In anaphase I, the homologues are pulled apart and moved to the opposite ends of the cell. The sister chromatids of each chromosome, however, remain attached to one another and don't come apart. In telophase I and cytokinesis, the spindle breaks down. Nuclear membranes then begin to reform around the groups of chromosomes on either side of the cell. The chromosomes then begin to condense back into messy chromatin. The cell pinches in the middle, forming a cleavage furrow, and 2 new daughter cells are formed. These 2 cells are now haploid, but still have replicated chromosomes. During prophase II, chromosomes condense again, and the nuclear envelope breaks down. The centrosomes move apart, the spindle fiber begins to form, and the spindles begin to reach out to the chromosomes. During metaphase II, the chromosomes line up individually at the metaphase plate in one line, similar to mitosis. In anaphase II, sister chromatids, not homologous pairs, are pulled apart by spindle fibers to either end of the cell. In telophase II and cytokinesis II, the spindle fibers disappear, nuclear membranes form around each set of chromosomes, and the chromosomes decondense. Cytokinesis splits the chromosome sets into new cells, forming the final products of meiosis, 4 haploid cells in which each chromosome has just 1 chromatid. In humans, the products of meiosis are sperm or egg cells.